This is going to be a short lecture on gene regulation in prokaryotes using what are known as prokaryotic operons. Now first, prokaryotes and eukaryotes are quite different in how they regulate their genes, meaning in how they turn on or turn off their genes and how they carry that out. On the left-hand side here is shown prokaryotes, and again they're going to have circular DNA shown there. But if you were to have, for example, a particular process, for example, maybe the particular bacteria will need to form the amino acid tryptophan. In synthesizing tryptophan, there may be many enzymes that are needed to carry out tryptophan synthesis from start to finish, because every basic reaction in that series of events to form tryptophan is going to need its own enzyme. Now, what a prokaryote will do is that all the genes necessary for all the enzymes that are going to be needed for tryptophan synthesis are all located together. You transcribe all the genes together. Here the genes are represented as the letters here. So A, B, C, D, and E. Here E, D, C, A, B, A. But here, those are all the enzymes that are needed for tryptophan synthesis. They are all transcribed together, so you get one long messenger RNA, and then that single messenger RNA is used to create all the different enzymes. So then you end up with enzymes EDCBA. Then they can go, all those different enzymes can then go create tryptophan. Now, that's how it would occur in a prokaryote. This is an example of an operon. Now let's compare that to what happens in eukaryotes. In eukaryotes, if you are going to synthesize tryptophan, the genes here numbered 1, 2, 3, 4, 5 are all on different chromosomes. And this is in a yeast. And so what would happen is transcription would happen separately for every single one. You would have, in this case, 5 different transcription events. One event creating a messenger RNA for number 1, a separate event creating the messenger RNA for number 2, and so forth. Then you would translate each of those separately, and you would still get the 5 different proteins. But in order to do that, you had to create 5 different transcriptional events. Okay. So in eukaryotes, it's very different. Even if all of those particular proteins, 1 through 5, were all needed together, they are regulated and transcribed differently. Okay, they are all transcribed uniquely, independently of each other. That's what makes the operon different. All the genes that are going to be needed together are going to be transcribed together, and that's what makes an operon. Now here is an example of how the tryptophan operon works. Again, the point of this is for the gene to make tryptophan. Okay. Now, the gene will have a promoter, which is up here, and within that promoter is a special region called an operator, and that's shown here in green. Now, what will happen is promoters with that operator, when in a bacterial operon, will be regulated by repressors. Now, in this particular case, if we look over on the left-hand side, there is an inactive repressor. Now think of just the part of the term called repressor. Repressor would mean stopping or decreasing. So if it's a repressor, the job of that little green protein there, if it was in position or if it was doing its job, would be to repress transcription. But as we can see here, it's inactive. So it's not repressing. So in this particular case, the gene is considered on, and you are going to transcribe. You're going to be carrying out transcription, and you will end up with the messenger RNA. So you end up with the messenger RNA. Now, if you look at the right-hand side, the repressor is now active. Well, what made it active? Over here, the little molecule in red is called tryptophan. So tryptophan, when bound to the repressor, activates it. Now, why does that make sense? What was the purpose of this particular gene? Why did you want to transcribe this particular gene? Well, it was to create tryptophan, that exact same molecule. Well, here, presumably, the bacteria was creating tryptophan for a particular purpose. So it had a job to do. So the tryptophan that was created initially was going to go off and do that job. But now there's just extra hanging around. And if you have extra hanging around, that means you have enough tryptophan. So now extra tryptophan will bind to the repressor. And now that means you don't need more tryptophan because you already have extra hanging around. So now the active repressor will bind to the operator portion of the promoter and turn the gene off. In this particular case, you no longer need tryptophan anymore. So the molecule that was going to be produced by this gene helps to shut it off. Now there is a separate operon, which is known as a lac operon. Lac here stands for lactose. This is a sugar that's found in, for example, milk. Okay. Now, lactose is an energy source. Think of it as a sugar, okay? And so bacteria can use it, ingest it, break it down, and use it for energy. Okay. Now, the lac operon also has a repressor. But its repressor acts a little bit differently because lactose is a little bit different than what we were doing with tryptophan. Remember, with tryptophan, you were building tryptophan. Now, with the lactose operon or lac operon, you are going to be degrading or breaking down lactose. You're not building it, okay? So lactose in the environment is going to be used. So in this particular case, the lac operon has a repressor that's always going to be active except in the presence of lactose. Now, why does that make sense? Okay. Well, the lac operon is going to be creating the enzymes needed to break down lactose. If there's no lactose around, then what's the point of making the enzymes to break down lactose if there's nothing to break down, if there's no lactose around to break down? So in this particular case, the repressor is only going to be inactivated in the presence of lactose. Okay, so another way to say that is the repressor is active unless you have lactose around. Okay. When present, lactose will bind to the repressor and inactivate it. Again, lactose here, allolactose is a version of the lactose. And here we can use them kind of interchangeably. Now there's a little bit different with lactose as well. It's a little more complicated operon. Glucose is the preferred meal. If it's going to break down something for a meal, bacteria would prefer to use glucose than lactose. It can use lactose, but it prefers glucose. So given the choice between the two, if both glucose and lactose are around, it will always choose glucose. So think about it in that case. If glucose is around, the bacteria aren't really going to want to use lactose even though it might be available. It may be the same as, for example, if you came home and you had a choice between 2 meals for dinner after a hard day of work and school. All right, so you have 2 choices for dinner. One of them, you'll eat it if you have to, but it's really not your favorite. And then the other choice was your favorite meal. How many times would you actually go for the one that was, I really don't like that? Okay. Most people would always go for their favorite. Okay. The bacteria are similar to that, so they will use the glucose as long as they have that option available. Now, there needs to be an indicator to the bacteria when glucose is not around. Okay? And there's a molecule called CAP that's going to act as what's called an activator that's going to detect when glucose levels are low. Okay, this is a little bit of a complicated figure, but if you take it into pieces, it will actually simplify itself a little bit. Okay. All right, so we have 2 different things here. First of all, represented in green is the CAP. This is the activator molecule. This is only going to become activated when glucose levels are low. So unless glucose levels are low, this is not going to be activated. What is it activated by? Cyclic AMP. Okay. So once cyclic AMP is around in higher quantities, it will activate CAP, and that indicates that glucose levels are low. The other thing we have is the repressor. Now remember, the repressor is always going to be active unless lactose in the form of allolactose is present. All right, so in our first condition, which is A here, here are the conditions off on the left-hand side for all of these choices, A, B, and C, are going to be given the conditions. In this one, you have no lactose, that's why there's a minus sign. You do have glucose, and remember, the presence of glucose means low cyclic AMP. And so those are the conditions. So if I were to ask you on a test, under those conditions, what would the lac operon be doing? Well, first of all, you should be saying, well, nothing. Okay? No messenger RNA transcription. And then if I asked you why, you would have to say, well, you need lactose present to inactivate the repressor. So here we have an active repressor. With an active repressor, no messenger RNA is going to be formed. So in this particular case, no lactose, no messenger RNA transcription. In addition, glucose is around. So it's going to want to use glucose even if lactose was around anyway. Now that brings us up to the second condition. Okay. In this condition, we have lactose, but we also have glucose. So the difference between A and B is we now have lactose present. Okay. So in this particular case, notice we now have an inactive repressor. It was inactivated by the presence of lactose. But because you have glucose, and I can even put that in red there, because you have glucose, the CAP molecule is not activated. So you will get transcription, but it's at very low levels. Now the last condition, over here. Now you have high lactose, but no glucose. So in this particular case, we have an inactive repressor due to the presence of lactose. But now, because the glucose levels are low, we have high cyclic AMP that activates the activator, which is called CAP. So we activate an activator. And now we get high transcription. This is the only case in which a significant amount of transcription would occur for the lac operon. Now please go back and make sure you understand why you get the different levels of transcription under the different cases. And please make sure you understand the role of lactose versus the role of glucose. What I'm going to do now is end the operon portion of the lecture. So this will be a short lecture presentation, and we will pick up translation on a different presentation.